Alright, now what I've done here is I've taken this sequence, this GPX 8 Homo sapiens, and what I did is I put it into BLAST and I searched against only human. In other words, this is a human sequence, alright, and I put it in, I uploaded the file into BLAST like we did in the previous assignment, and but I chose human, so the same thing, that the same organism that the sequence comes from, I searched essentially the rest of the human genome using that sequence mainly to see what is similar and how similar there are other things within the human organism, within the human genome, that what other things are similar to the GPX8 gene. Now you would expect it to be the other GPX genes, but you have to search just to make sure to see what you get. So here are the results from that search. Now as you can see, I get 100% on both of these plus a 0 e-value, which means this is the actual sequence that I used, that I searched with. And then here are the other things. So here we learn something. We learn that there are variants of the GPX8 gene. These are probably spliced variants, so you splice them different way, and if you were to look at these, you would see that splicing is different. The other thing is transcription might start at slightly different places. Those are the two ways that you can get variants. There could also be mutants, but these are probably variants in the sense that they are spliced, and perhaps transcription start site alterations are also possible. Probably not mutants though. But notice this says non-coding RNA. If these are non-coding, then these are pseudogenes, and they might be involved in RNA interference that we talked about in class. So if you look through these and realize these are predicted, that means they've never actually found the RNA in when they like took cells and break them open and look to see what RNAs are in there. So they've never actually found this RNA, but they predict that this is it. That's why you say predicted. And that's kind of what we do. We look at the sequence and then we kind of predict where things are going to go. That's all we can do in here. But of course you have real laboratory studies where they extract the RNAs and sequence them. So this is really, we learn a lot from just looking at this. We realize that there are, GPX8 has 8 variants, if not more. I just see 8 here. So we could analyze those if we wanted. I'm not interested in doing that, but that is like something that could be done. But what I'm more interested in is looking to see what genes are similar to it. So down here, we have this RNA. Let's see what this is. This is some kind of clone, probably from a larger piece of DNA or something. But I'm going to open this. I'm going to go over here. I'm going to click on this, and we're going to read about what this is. We're going to read here a clone from a sequencing project. So it picked up that part. Someone was sequencing the human genome, and they picked up this part of the genome, I'm sure. I doubt that this is a duplicate. Alright, now we scroll down. More clones, cDNAs, another glutathione. Okay, now here's something a little different. Homo sapiens BICRA-like chromosome remodeling complex protein. This does not look like GPX here, and it has 90% identity. It still has a really high e-value, but not so much coverage. So there's a piece here in whatever this is that's very similar. We've got dogs barking in the background, if you can hear them. But so this is interesting. So we find that there is something that has quite a bit of similarity to GPX8. Go back. that would be interesting and worth looking at. Oops. Moving forward. Okay. So let's click on this one here and see. Let's just look to see how big of a FASTA that is. That's considerable. So it might be that this is a larger piece of DNA and it just happened to get incorporated into it. Uh, Complex associated protein, BICRAL. That might be, it's a remodeling uh, complex. In other words, this is when you want to change how the uh, DNA wraps around the histones. We talked about that. So this is interesting. It looks like it's on a different chromosome also. So this is actually a pretty interesting thing. This gives you some hints as to uh, maybe GPX has multiple functions. It very easily could be that GPX 8 uh, is also involved in chromosome remodeling, which means moving your DNA on and off of the histones or to a different set and rewrapping the DNA, which might, of course, be something like turning it on and off. And since this is primarily found in the brain, I would say that's probably true. It's probably some type of mutation that led to the formation of GPX8, um, probably relatively recently in humans. But that's all speculation. I can't prove any of that. We'd have to do a little study. But the idea is you can learn a lot about a sequence by looking to see what it has similarity with. And so when you look down here, we know, of course, that it's going to be similar to GPX genes. But before we even get to like GPX 7 or 6 or 1 or whatever, we're finding other genes in here that have similarity to it. And that is very interesting. So what I'm going to have you do is I want you to do exactly what I'm doing here. And I want you to find a few of these that are not GPX genes, all right, like I'm doing here, and download these and find out where they're similar and see if they are, how they relate to, if they're in the same places of the ones that when you're comparing the GPXs across the genome. In other words, so for this one, this one's a little long. I'm not gonna, you can pick whichever ones you want. You might look for some smaller ones, but it would be interesting to find out what pieces here are similar to these other genes and see how those match up with your previous assignment. So your next assignment is to do as I've done here and go through and find some of these pieces within the same genome that are not GPX genes. So we don't want to know what this is similar to with regard to GPX. We want to know what this is similar to outside of the GPX genes. So this will be your next assignment, and I'm going to say pick 5. So if you were to get this result, go through, read them, and if there's something, don't choose things that are clones. Get something that actually has a name, like this was the BICRA, like chromosome remodeling complex. That sounds like it's something in particular that does something, and this is actually a set of genes that look like they go together. This is a little long, so if it's a little long, maybe you find something else. This is a clone, an isolate. You might try something like this. Let's see what this looks like. How long is this thing? Let's go to the FASTA. That'll tell us how big it is. Another pretty long one, probably not something we want. It might just be a piece of DNA that includes the GPX gene, so that's not what we're looking for. We want to find something. Oh, here's a tRNA splicing endonuclease. In other words, this sounds actually very interesting. Let's look at this. All right. It's still it's a long sequence, but that one sounds interesting enough. I would probably do that one. But the problem is these longer sequences probably include DNA that includes the actual sequence. So one way to make sure it doesn't is to find out what chromosome it's on and see if it's on the same chromosome as your GPX. And if it is, then don't use it. It's probably just it's a sequence that includes yours. But if it's not, and this is, I don't believe GPX 8 is on 3, so this would be an interesting one to look at. But it's still a bit of a long sequence to be messing with. I don't want to slow your computers down. These long sequences um, can be difficult on a laptop. Here's one from chromosome 16. So go through, and what I'm going to do is I'm going to assign you to find 5 of these, and all I want you to do is report the numbers over here, the query and the e-value and the percent identity. Report those, look at them, do a dot plot, and see if it's the same region that you've seen before. See what regions are matching up, and I suspect they are not the same as when you compare your two GPXs. So you compare your two GPXs, you're going to find things that are specific to GPX genes that have to stay the same for them to maintain their function. But here you're going to find other pieces and lead to other clues as to maybe how these genes are regulated. So this will be the second assignment for the week. Do the first one and then try this one, and you will report on the 5 genes that you pick. You can pick them. I don't care here, like thymidine kinase, and actually here it shows you. But is this the same region? Are these the same locations as the similarity between GPX? It might be, but probably not. All right, let's see here. And that's your next assignment.